So we have a phosphate and a sugar backbone that comes along our DNA strand. So as you can see on the sugar itself, there's 5 carbons, and the carbon 1' is connected to our thymine. That thymine is also connected by a hydrogen bond to our adenine. And as you can see, the orientation of the DNA strand is read from 5' all the way down to 3', and you can see that the opposite strand is antiparallel, meaning that the 3' is aligned with the 5', and it's read backwards almost, so 5' all the way up to our 3' that is not labeled.
So with the semi-conservative model for DNA replication, essentially one DNA molecule, both strands split, and then when they do split, complementary bases bind to the single strands that do split, and then they reform again to form 2 separate DNA strands, and then they just keep doing it until there's enough replication of the DNA.
So in our DNA, our double helix is split in a DNA fork. So basically from 3' to 5', our DNA becomes discontinuous, and then from 5' to 3', it is continuous. And so basically what happens in the discontinuous portion of the DNA, our new strand is split up into Okazaki fragments, and so we have what becomes 5' to 3' in order to complement the opposite side of the DNA, and this is going to be sealed by DNA ligase, whereas with our leading strand from 5' to 3' or 3' to 5', our opposite strand is going to complement the nucleic bases. That's not right, is it?
Nucleotide?
Nucleotide basis. Yeah.
So the reason why why linear DNA shortens with each copy is that if you look, there's an RNA primer on the 5' end which can be removed, but it can't be replaced with DNA because there's no such thing as a DNA polymerase that can attach to the 3' end of the DNA strand. So as a result, with each successive copy or replication of DNA, it gets shorter because of that lacking DNA polymerase. And as for why bacteria don't need to worry about this, is that the chromosome and DNA of bacteria is circular, so there are no ends to it.
So as you can see, our DNA is a double helix, and a bunch of histones will actually attach themselves to the double helix. This double helix then compresses itself, and it makes this chain that is called a chromatin. The chromatin itself, once it's condensed into a scaffold or a bunch of loops, it becomes a chromatid, and the replication of the chromatid, both of them together actually make a chromosome. So basically, depending on how the chromatin itself is packed will determine how easily the genetic information can be expressed. So if it's loosely packed, it's called achromatin. Euchromatin. Thanks. And so that's loosely packed, and that's easier for the gene to be expressed, whereas when it's tightly packed, that's called heterochromatin, and that makes it much more difficult to express.